Do you ever wonder what this whole genomics thing is all about? Are scientists using gene editing to create the next Jurassic Park? And what is this whole CRISPR thing I keep hearing about? We'll answer all of this and more on our second episode of Things That Make You Go, Huh?
Things That Make You Go, Huh?
So today we're going to be talking about CRISPR, which is without an E. I thought it was some type of crispy fried type of food, but actually it is something more scientific. So I brought in my colleague today from AbbVie. His name is Chris Miller. Chris, would you like to introduce yourself and tell us a little bit about yourself, very briefly?
Sure, yeah. So hi, I'm Chris Miller. I work here at AbbVie in the Genomics Research Center. We do a number of things related to using genetics and genomics to help drive drug discovery and drug development. And one of the tools we use a lot in the lab is CRISPR, which we're going to talk a lot more about in the rest of the podcast.
The reason I kind of thought about this in the first place, it's kind of a funny story, but a friend of mine recently asked me if I could take them to the post office. So I was like, yeah, sure, I'll take it to the post office. Well, then she proceeds to pull out a little test tube, and then she spits in it, right? So I'm like, what is going on here? And then she sticks it in an envelope and mails it. And so she told me she was mailing it in to do some type of DNA analysis. And so I guess it brought me to the thing of how can spitting in a cup tell my friend anything about possible health risks? So perhaps maybe you can shed some light on that topic before we kind of get into the main one.
Sure, yeah. So what your friend was doing was spitting into a tube so that it could be then sent to a company. There are a small number of companies that do this. Actually, a fair number of companies, but several that are actually credible and experts and do a really good job at this. And what they do is they will take that saliva sample that you spit into a tube and extract DNA from it, and then analyze what are called variants, or SNPs, single nucleotide polymorphisms, and sequence variants across the DNA, and try to make some inferences about some health risks and ancestry information. And
they can get that just from a little wad of spit, basically.
A little bit. It's a couple of mils, a small amount of a couple of teaspoons maybe of saliva that's in the tube, and they can extract DNA that's either in the saliva or comes from the cells on the inside of your cheek, and you can extract DNA from it, and that's enough to do the types of analysis that they do.
Okay, so let's start off small here. I figure we'll start off small before we get big. I was horrible in biology in high school, so can you just, for the layman, tell me, for me and for the lay people out there, what DNA is?
Sure. DNA is a substance that's inside every cell in our body. It's in the nucleus of every cell in the body.
Is the nucleus the center of a cell?
The nucleus is the center, the nucleus, the center of the cell.
Okay.
And that's where all the genetic information is, or the vast majority of the genetic information is. And DNA is made up of 4 different chemical molecules that are strung together into what are called chromosomes. And every cell in your body, again with a few exceptions, has 23 pairs of these chromosomes. And they basically, you can think about them as books. And if the entire genome that represents all of your DNA is a volume of many books, each chromosome is a single book, and they can be different lengths. But within those books are the letters of DNA. So in the same way that there's words and sentences that are in a book, there are sequences of those letters that are strung together that you could consider to be words and letters. And
so everybody has their own story?
Everybody has their own story, although in terms of the similarity across people, more than 99% of my book is the same as your book, is the same as everybody's book in this room.
And are the books, if we're using a book as a metaphor, is that like more similar within my own family?
It will be more similar within your family and those people that you're most directly related to. But the books and the words, they're made up of just 4 letters. So, in a normal alphabet in the English language, we've got 26 letters. But in the language of DNA, there's only 4 letters. And they're referred to as A, G, C, and T, or adenine, guanine, thymine, and cytosine. And it's the exact sequence of those over and over again in endless permutations that make up the sequence of the DNA. And within each cell in your body, again, you've got 23 pairs of chromosomes. You have roughly 3 billion of those A's, G's, C's, and T's. And if you actually string them all together, the amount of DNA that's in each cell of your body, it's about 2 meters long. And it's all packed into the nucleus of each cell in your body. And you've got more than 30 trillion cells in your body. And it's all packed in there in a very structured way. It's not just randomly jammed into this tiny space. And it's got very important consequences about how this all works. And it's actually really fascinating in the way that it translates into biology and disease. Both the sequences, the way that those A's, G's, C's, and T's are strung together, and then the way it's all packed together.
This is going to sound like an odd question. Are those created upon conception, or do they grow as you grow? Do you know what I mean?
It's the same DNA at conception, when egg and sperm combine to create the 23 pairs of chromosomes. But it's the same DNA with a few exceptions, but throughout life. And I don't want to get into the exceptions of it, but things like sperm and egg, they don't have 23 pairs, they just have 23 single chromosomes. And there's a few other cases where it's not that, but by and large it is.
Oh, that's cool. What about RNA? What's RNA?
So RNA is chemically very similar to DNA. DNA actually stands for deoxyribonucleic acid. RNA is actually ribonucleic acid, so it's just a different chemical structure, but it's got a lot of similarities to it. It's largely based on the same AGCT concept, although in RNA the T's are something called uracil. They're slightly different chemically. But again, they're all strung together in a way that means something, and that the exact way they're strung together, they tend to be, they encode for what are called proteins. So there's this thing called the central dogma of molecular biology, and this has been around for many, many years, decades, back into the early part of the last century. But the basic idea is that you have DNA, which has these A's, G's, T's, and C's strung together. And there's actually two strands of this that go in opposite direction, and they're connected together like a ladder. And one strand determines what the other strand is. They're mirror images of each other in some ways. And so what happens, so DNA is this deoxyribonucleic acid, this double-stranded helical structure. RNA represents one copy of that. And there's ways that inside the nucleus that it's copied into this one copy of it. And then RNA actually gets what's called translated into protein. And protein is what you see. It's the structure and the function of the body. So when you look at your hand, what you see largely in skin is keratin. It's a type of protein. But there's many other proteins that are part of the body that are the structural pieces of cells, the inner workings, the machinery, enzymes. The way that cells work is all about proteins. So there's this central dogma that goes from DNA to RNA to protein. And there's, again, a few exceptions to that, and as we learn more and more about it, it gets more complicated than that. But that's the basic idea. The information in the genes made into RNA, made into protein.
Okay, I'm a little lost with the RNA, but I understand the DNA part.
Think of it just as the intermediary. It's
okay.
so
is RNA created when DNA is created? Are they created in conjunction with one another?
No. Well, no. The DNA is created when cells divide. The DNA that's in your cells gets copied and then split and then goes into new cells. So that's how DNA is made. Copies of itself are made and then put back into the nucleus of new cells. But the RNA is a molecule that's made by a different set of enzymes and proteins that give you this messenger intermediate. So it's similar chemically to the DNA, but it's got a different function, and that is to give the instructions to how you make proteins.
Ah, okay. Okay, I think that makes a little bit more sense. So let's move on to what is a gene? Can you help me with that one?
Sure, yeah. So you can think about a gene either in conceptual or actual molecular and real terms. But the conceptual is it's the basic unit of heredity. And so that, you think about hair color, eye color, height, any number of attributes that we have. those are encoded by and controlled by genes. And if you look at your parents, there's a high likelihood that some attributes of yourself you can see in your parents. But it does determine, it's the basic unit of heredity, and it determines the things that are the physical things that we can see, and many of the things we can't see as well. So the traits that we have as people.
So like what? Like you said, you kind of mentioned before, like eye color, hair color,
height, male pattern baldness, any number of things that that we can see when we look at the number of fingers we have, the number of legs, and although in those cases it's not like a single gene that says we're going to get 5 fingers. It's many genes that work together to program and say that we are actually that's how many fingers we're going to have. But that's kind of like the conceptual level, the basic unit of heredity. But at the molecular level, the genes are regions of the DNA that we just talked about, a few minutes ago, and it's regions that contain the basic sequences that are going to get made into RNA that then get translated into protein, and all the stuff that's part of the genome region around that gene that regulates when it's going to be turned on, when it's going to be turned off.
I don't know if you've already answered this, but what is the genome?
The genome, it really just refers to the full complement of all the genes that we have, that any cell or organism has. I mean, so we speak about the human genome, and that's, again, about 3 billion of these A's, G's, T's, and C's. It's the 20 to 25,000 genes we have, and all the rest of the genome, the DNA, that makes it all work. And so, again, as far as the human genome is concerned, only about 1% of it is the stuff that gets made into mRNAs and then into protein. And only about
mRNAs?
Into mRNAs and then into proteins.
Yes.
And then about 25% of it is the stuff that regulates that 1%. So in truth, about 75% of the genome is, we either don't know what it does or we know that it has no function. And so most of the genome is not this stuff that is responsible for the making of new mRNAs and new proteins. It has other functions, whether it's structural or regulatory or things that we don't yet understand.
You keep saying mRNAs?
Messenger RNA.
Messenger RNA.
mRNA,
okay.
yeah, sorry. The M just means messenger. And so we talk about DNA, we talk about RNA, but usually what we're talking about with RNA is mRNAs or messenger RNAs.
So going back to the beginning of our discussion, how does my friend sending in her spit give her any type of information on her risk of disease?
So that's a great question. So there in some cases, there's actually a really good and strong case to be made where that is in fact possible, to be able to predict risk of disease. In many cases, though, there's not a strong case. So it's a very hard thing to do to make this really strong connection between a genetic mutation and disease or disease risk. And, we're just at the beginning of this whole revolution in terms of how genomics and genetics influences what we do here in drug discovery and drug development, and then moving forward, how it influences clinical practice. At a certain point, every baby that gets born is going to be genotyped to a sequence. Then at a certain point, in the not-too-distant future, every time anybody goes sees a doctor, your sample will be taken, your genotype, your entire sequence will be available to a doctor, and with the appropriate counseling and information, you'll make health decisions on it, which won't be just guessing in the dark, but they'll actually be based on real science.
Okay, I think you've given me a lot to think about and also a pretty good base for the basics here. Let's talk about gene editing. So you're talking about all of these traits and qualities of genes. Can you actually do something to the genes? Can you edit them in some way to somehow mitigate your risk of acquiring a disease? Does that make sense?
It does make sense. Short answer is today, no. At some point in the future, perhaps. The science around gene editing has actually exploded just in the past few years. There were some basic techniques for trying to make, and gene editing really is just referring to making changes in the genome. Editing, you think about editing a manuscript or a paper, you go in and you change things, hopefully make them better. Genome editing, genome engineering, or gene editing, it's all the same thing, is basically being able to try to make some changes in the genome. And so, again, there's been methods around for decades to try to do this, but they haven't been very efficient. They've been very labor-intensive or expensive, but a lot has changed just over the past 4 or 5 years. The technology that's referred to as CRISPR is the thing that's really changed that significantly.
CRISPR is gene editing?
CRISPR is gene editing. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's quite a mouthful.
Say that 10 times fast.
Yeah, exactly. So that's why they call it, they shortened it to CRISPR, CRISPR. It's a system that we can use to make these changes in the DNA. It's a system that evolved in bacteria as a way of, it's actually a bacterial immune system as a way of building immunity against viruses that were infecting bacteria. But we understand the basic machinery to do this, and now we can use this in mammalian cells to make specific changes in DNA. We do this a lot in our research group for what's called target ID and target validation, and we make models, disease models using CRISPR if we want to make specific changes. But basically, it's just a small set of proteins. One in particular, it's called Cas9, CRISPR-associated protein 9, it's a whole family of them. But it's an enzyme that can cut DNA. And when it cuts DNA, two things can happen. In some cases, the cut will just re-heal, if you will. But what happens is a couple of the nucleotides or bases, the A's, G's, C's, and T's, get nibbled off the end, and what you end up with is a very small deletion, or in some cases an insertion. They call them indels, insertion, deletion, but a very small change right where the cut happened. In other cases, if you can provide another piece of DNA in the right way, that other piece of DNA can get inserted into the cut. And so rather than just healing over, it heals, but now it's got something that's inserted in the place of it.
So let's just take a step back here because this is extremely confusing for me.
Yeah.
So CRISPR, is it like a technology?
CRISPR is a technology
Okay.
that lets us make these cuts.
Okay. And it has something to do with an enzyme? The enzyme is making the cut?
The enzyme is the protein, this Cas9 protein, that actually makes the cut. And that's what it does, and that's what it did in bacteria as a way of trying to cut DNA that was when viruses infected. But it's the enzyme that makes the cut itself.
And you're either just making a clean cut, or you're inserting something into it.
Basically, yes.
Okay.
And if you're trying to just make a cut, you can do it in a way that inactivates whatever you've cut. It's this molecular scissors concept where you're just cutting it, and what you've done is you're inactivated. You're stopping it and interrupting it so that it no longer functions like it did before it was cut.
And then when you insert, are you reactivating it, or no?
Well, you can, or you can just insert something that now represents a mutation, a small change that you're specifically trying to make.
So you're okay. So I'm just trying to see if I understand this. So you're cutting it to deactivate it, right?
Yeah.
But you can also, can you put it back together once you've cut it?
You can, but in general, we don't try to fix the things that we've cut. We will be asking a different question, where in some cases we don't want to just cut and inactivate. In some cases, we want to make a single change and study the consequences of that. So, so it's not so much that we want to repair what we've done. It's more a different question that we're using.
Okay, so you cut it to see what happens next. So you obviously put a lot of thought into why you're cutting a particular gene.
Yep.
You're cutting.
Yeah, although I'm mostly yes, but not always. In some cases, what we do is we actually have libraries of these CRISPR molecules, these Cas9 molecules, and actually the machinery that helps us do this, but libraries that cover all human genes. And we can set it up so that we do what are called screening experiments to say what genes, when we knock them out, when we inactivate them, give a certain response in a cell-based system or an animal model. So if we've got a cancer cell line that we have in growing in a petri dish or in an animal model or something like that, we can infect en masse this whole library of these CRISPR molecules, and in various ways that we have at our disposal in the lab, identify what genes are the vulnerable points that can, let's say, stop, if we inactivate them, can stop that cancer cell growing or change its response to a drug. So we don't have to a priori know this is the gene we want to inactivate. We want to identify the gene that when it's inactivated gives us the response that we're looking for.
And so let's talk about I'm calling it mutating one, when you put something in.
Yep.
What are you looking for when you're doing that?
In that case, it's much more of a targeted question where we do need to know ahead of time what it is, what's the question, what's the gene, and even what's the mutation that we're trying to make. And so one of the things that's a growing use case for us is that we are putting a lot of effort, mainly with outside partners, but a growing amount internally, on sequencing people who have different types of diseases, and we want to understand what are the mutations in those people that make them more likely to develop Alzheimer's or multiple sclerosis or Crohn's disease or a disease called axial SpA, for example.
That's pretty cool. I mean, it sounds, sorry to interrupt you,
that's okay.
It sounds like a very powerful tool.
Well,
right?
so the
If you use it
sequencing,
correctly,
so absolutely. So I mean, there's several powerful tools here.
So this may sound controversial, but so can you manipulate? It's almost like you can manipulate genes in essence, right? So we were joking outside of the podcast about, can you like create another Jurassic Park or something? Do you know what I mean? Can you create a dinosaur?
Today, no. Certain people believe it can be done, and I think many years, if not decades, from that being done. But dinosaurs is another. We're talking millions of years to go back. They're tens of millions of years, and the likelihood that we'll be able to recover intact DNA from dinosaur bones.
There's not like dinosaur spit hanging around.
No, not that we know of. But I mean, just the complexity of doing it too. How would you do it to make dinosaurs? Or the Jurassic Park was, what did they put them into? Was it frogs or some kind of reptiles, little pieces at a time? And, and of course, we know how that turned out.
It's still turning out. There's still sequels coming out.
Yeah,
so.
that's true.
So what would you say makes this technology so revolutionary, or why are people so crazy for CRISPR?
Well, so a key thing is it just enabled us to do what we had always wanted to do before in a much more efficient way. And like I said, there were technologies before CRISPR that we could do these, we could make changes, we could inactivate genes, but again, very expensive, very slow, and just not amenable to the types of things that we can do now. I mean, the genome-wide screening that I was mentioning earlier, there were some earlier technologies that moved in that direction, but CRISPR made it possible in a way that just wasn't previously possible.
So Chris, kind of coming to the end here, I'm just wondering, if you imagine what CRISPR can do in, let's say, the next 5 to 10 years, where do you think this technology can take us, and what is your hope for what it can accomplish?
It's come so far in such a short period of time, and the trajectory we're on is actually quite remarkable. I've never seen a technology evolve this rapidly and with this widespread use and improvements and so on. So in the research arena, I see continued evolution of new things that we can do with CRISPR. Initially, it was just about trying to inactivate genes and maybe make some of these mutations that I was talking about earlier. But there's some really fascinating and interesting new variations on the theme that are changing it's not just about cutting DNA, but about changing DNA and answering some questions that we just wouldn't have been able to answer before. So I see some revolutionary things coming in the research arena for CRISPR. And on the therapeutic side, I think we're kind of at a tipping point where there's a number of companies that have some ongoing clinical trials that in the next year or two are going to either work or not work. And I hope they're going to be successful. They're largely focused on genetic diseases and some limited delivery to places like the eye. And if those are successful, I think the floodgates open for new applications, new diseases, new delivery methods, and I think the sky's the limit. So I think we're at a tipping point for therapeutics, and I think the next year to two are going to be really exciting.
Okay, so thank you so much, Chris, for your time today. I really appreciate it. And this stuff is really hard for me to absorb, but you actually explained it in a way that I think I kind of basically understand it. So thank you for that. You achieved miracles today, Chris. So, and since it's the end of the podcast, I do have to put my money where my mouth is and try to describe some of the things that I learned today. So I'm going to do my best here to summarize kind of some of the stuff that you talked about today. So I guess the first thing is that I think that I got from today is that DNA is some type of genetic material that basically makes us who we are. Does that sound okay?
Yep, so far so good.
RNA is kind of the intermediary, I guess, between the DNA and the proteins.
Yep, 2 for 2.
Okay, so with a technology called CRISPR, we can either delete a section of a gene or add some type of mutation to that gene.
Yep.
CRISPR is extremely revolutionary because, it can allow us to see how changing genes one way or another can basically change the course of a disease.
Yep, that's true.
Oh my goodness, 4 out of 4 here. And last but not least, there's no need to worry about Jurassic Park, at least in the near future.
For now.
For now, right? So I can sleep better at night. Thank you so much again for coming today, and look forward to seeing us next time on Things That Make You Go, huh?
Things that make